Hello everyone, thank you for attending today's webinar about our LNP platform. I'm going to launch the presentation. It was pre-recorded by our colleague Kendall in the US for a better understanding. Please stay until the end because we will have a quick question and answer session. And then we have also a satisfaction survey, so if you can fill it to help us improve, it will be great. I'm going to launch the presentation. If you have any question during the webinar, do not hesitate to ask.
OZ Biosciences is a French biotech company founded more than 20 years ago, specializing in molecular delivery systems present in both France and the US. The French site is located in Marseille, comprised of the headquarters and the production site, divided into 2 laboratories over 500 square meters. OZB SAS is responsible for R&D, production, mRNA and LNP platforms, marketing, and sales. The US site is located in San Diego and deals with marketing, sales, and business development. Over the years, we have established a worldwide distributor network. One of our objectives is to develop the spread of our reagents and expertise. This expertise is built on more than 20 years of experience and innovation. Several important milestones have marked the company's history, such as the commercialization of the first magnetofection reagents in 2004, the creation of OZB Incorporated in 2013, or our recent acquisition of new facilities for the development of the mRNA and the LNP platforms. We are proud to cover a wide variety of fields, from bioproduction to viral applications, through in vivo protein delivery or stem cell applications. All the products and platforms are developed and sold under ISO 9001 certification. These innovative reagents allow OZ Biosciences to offer solutions in 3 main areas: cell and gene therapy, bioproduction, and custom platform productions. Today, we are going to focus on our LNP production platform. OZ Biosciences offers a comprehensive LNP solution ranging from ready-to-use catalog products to fully customized formulations tailored to your specific application and cargo. Recently, we have implemented targeting capabilities to our platform that we will present to you later. Our LNP production platform is built around a streamlined and well-controlled 4-step workflow. From design to release, it takes approximately 2 to 3 weeks, depending on the project. Step 1 is LNP design with the selection of the lipid composition and definition of the cargo, which can be any nucleic acid or small molecule. When considering mRNA as cargo, our in-house mRNA production platform can synthesize it for you, ensuring full quality control from sequence to final encapsulated product. Step 2 is LNP manufacturing. This includes lipid preparation and the critical microfluidic mixing step that controls particle size and encapsulation efficiency. Step 3 is the downstream process, dialysis and buffer exchange to remove solvent residuals, exchange buffer, and concentrate the product. Step 4 is quality control. Size, zeta potential, encapsulation efficiency, and sterility are some of the parameters that are checked. Our LNPs are classically composed of 4 lipid components in defined molar ratios. The ionizable lipid at 40 to 50 mol%, which drives encapsulation and endosomal release, the helper phospholipid at 10 to 20 mol%, which contributes to membrane stability and fusogenicity, cholesterol at 35 to 45 mol%, which rigidifies the particle and supports membrane fusion, and a PEG lipid at 1 to 3 mol%, which stabilizes the particle surface, limits aggregation and nonspecific interactions, and controls blood half-life. Particle size and zeta potential are fully controlled by adjusting the lipid ratios, as well as the mixing conditions by microfluidic or jet mixing. Targeting agents such as peptides or antibodies can also be conjugated to the surface. This composition is compatible with any kind of nucleic acid cargo, such as mRNA, saRNA, or DNA, as well as small molecules. As we like to say, we master every step, component, and formulation of our LNPs. The ionizable lipid is the central component of an LNP. It is responsible for nucleic acid encapsulation, endosomal escape, and ultimately intracellular delivery. OZ Biosciences has developed a library of more than 50 patented ionizable lipids. These ionizable lipids are unique due to the presence of groups inspired by the structure of amino acids, which significantly enhance their biocompatibility. This bond, which can be cleaved in a biological environment, gives them a high level of biodegradability. The architecture of our ionizable lipids is highly modular. Several structural parameters can be tuned. The head group, which determines the charge and aqueous interactions, the linker region, which mimics amino acid structure and confers biodegradability, and the hydrophobic tail, which can vary in chain length, degree of unsaturation, and branching. This structural diversity gives us flexibility to fine-tune delivery efficiency, biodistribution, and tolerability for each specific application. Before any LNP product leaves our facility, it goes through a series of physicochemical quality controls. We systematically measure particle size by dynamic light scattering. We target a size typically between 50 and 200 nanometers, depending on the LNP or its application, with a narrow size distribution reflected by a low polydispersity index. We also measure the zeta potential, which reflects the surface charge of the particles and helps predict colloidal stability, or encapsulation efficiency, assessed by a fluorescence-based assay, always above 80%. These parameters together ensure that our LNPs are well-defined, stable, and ready for downstream biological evaluation. To illustrate this, here is a real example from an mRNA-loaded LNP batch at 0.1 milligrams per milliliter. The size distribution shows a single peak at 157 nanometers with a polydispersity index of 0.097, demonstrating monodisperse homogeneous preparation. Zeta potential is close to neutral, which is the expected profile for these pegylated LNPs with a charge equal to 1.88 millivolts. Stability is a critical parameter for our LNPs. We confirm the stability of our LNPs over 6 months under various storage conditions by measuring physicochemical parameters such as the size or biological effect with luminescence reading. We can see that over a short time period, up to 2 weeks, size and biological effect are not affected by the various storage conditions. And when LNPs are stored at minus 80 degrees Celsius for a longer period of 6 months, both size and biological effect do not significantly vary. For short-term storage, 4 degrees Celsius can be used, and minus 80 degrees Celsius is recommended for long-term storage. This stability profile ensures that our products can be shipped and used reliably over a long period of time without compromising delivery performance. Let's now move on to our catalog LNP. We propose various categories. mRNA LNPs for in vivo organ selective delivery, multi-organ, liver or spleen targeted, as well as in vitro delivery and immunization. Considering in vivo LNP, the different tropism is achieved by varying the ionizable lipid and not active targeting. siRNA LNPs for gene silencing are available for multiple control genes, GFP and GAPDH, or LNP encapsulating Sci-Fi labeled non-targeting siRNA. We also propose a DIY kit designed for researchers who want to develop their own formulation at different N-to-P ratios, and also fluorescent LNP for tracking cellular trafficking. Finally, our selection kit, which lets you screen 3 or 5 different formulations to identify the one that works best in your model. Our ready-to-use LNPs are available with either our proprietary lipids or commercially available ones such as SM102 or MC3 and come in different categories. We have validated our LNP in vitro across a broad panel of cell types using GFP mRNA as reporter cargo. The graphs show the transfection efficiency in some cell types such as dendritic cell lines or Jurkat T cells using different LNP formulations at concentrations and compared to REM-spec transfection as positive control represented by the black bars. Results are summarized in the table on the right with the cell types tested, their origin, and the ionizable lipid we recommend for each application. We have demonstrated efficient transfection, for example, in B cells and T cells, as well as common cell lines such as HEK293 and CHOK1, or primary cells, including PBMCs and T cells from different origins. These results demonstrate the versatility of our lipid library. We recently launched a new formulation dedicated to stem cell transfection that encapsulates GFP mRNA. LNP stem cells allow for the transfection of primary CD34 positive cells with greater efficiency than SM102 LNPs, as shown here in the two graphs depicting the percentage of GFP positive cells and the average intensity for different amounts of mRNA. Regardless of the dose used, this formulation does not affect either cell viability or the CD34 positive phenotype, as demonstrated here by CD34 positive staining. Moving now to our LNP designed for in vivo delivery, we use luciferase mRNA as a reporter to assess biodistribution and expression in living animals after intravenous injection. The LNP Multi-organs encapsulating mRNA Luc shows a broad luminescence signal across all tested tissues such as lung, skin, or pancreas, with expression confirmed by both in vivo imaging and by ex vivo organ quantification. This systemic distribution makes this formulation particularly suited for applications requiring simultaneous expression across tissues. Using our liver-optimized formulation, we achieved a highly localized bioluminescence signal in the abdominal region in vivo, confirmed by ex vivo imaging of isolated organs. Quantification shows that the liver accounts for more than 70% of the total luciferase signal, demonstrating a strong liver tropism. Importantly, this liver targeting efficiency is superior to SM102 LNP, a commercially used reference ionizable lipid. With a spleen-selective formulation, we demonstrate highly concentrated bioluminescent signal in the splenic region in vivo, validated by ex vivo bioluminescence imaging. Quantification confirms that the spleen accounts for more than 90% of the total luminescence signal that reflects an excellent splenic tropism. Again, this performance exceeds what is obtained with SM102 LNP, reinforcing the advantage of our proprietary ionizable lipids for immune organ targeting. Let's now look at our LNP for immunization that encapsulates OVA-encoding mRNA. LNPs were delivered by subcutaneous prime boost injections on day 0 and day 14 in C57 Black 6J mice. The results show a strong humoral immune response with high anti-OVA IgG1 and IgG2C antibody titers in the same range than known vaccine adjuvants, reflecting activation of both T1 and T2 pathways. We also observed a robust cellular response with strong IFN gamma and IL-18 production, markers of T cell activation. These results confirm that our LNP immunization is highly effective at priming a strong and balanced immune response, making it a relevant tool for mRNA vaccine development. Our LNPs are also being used as reference standards in a recent publication by Limitsu et al. In 2026 in the Journal of Lipid Research. Our empty and mRNA-loaded OVA LNPs were used to investigate the inflammatory mechanisms underlying mRNA vaccine reactogenicity. 0.125 micromole of empty LNPs and 5 micrograms OVA mRNA LNP were injected intramuscularly in mice. The authors identified an omega-3 fatty acid metabolite as a potent mitigation strategy. It significantly reduces local swelling and immune cell infiltration, as shown by the red dots, without affecting OVA-specific antibody production. This study validates our LNPs as a reliable experimental model, and it also highlights OZ Biosciences as a partner at the intersection of lipid biology and vaccine science. We'll now move on to our custom LNP platform. We work with both our proprietary ionizable lipids, for which licenses are available, and with commercially sourced lipids. depending on your regulatory and scientific requirements. We can functionalize LNPs with targeting agents, peptides, antibodies, and other ligands. In terms of cargo, we can encapsulate any type of nucleic acid such as DNA, mRNA, siRNA, and as well as small molecules. We offer fluorescent labeling of both lipids and nucleic acids for tracking and biodistribution studies. Particle size and zeta potential are fully characterized, whether for in vitro screening or in vivo administration. And our manufacturing scale ranges from 0.5 milliliters to 1 liter, accommodating everything from early stage feasibility work to larger preclinical batches. We can support every stage of your mRNA LNP production, from mRNA synthesis to LNP formulation, development, manufacturing, and fill and finish. Let me share a recent example of what our custom LNPs can enable in the context of fundamental research. In a publication from Jager Mediot et al. In the Journal of Experimental Medicine in 2024, our LNPs were used in a sophisticated microfluidic lymphoid organ chip model to assess mRNA LNP capture and expression by myeloid cells. We provided control LNPs formulated with a Moderna-like lipid composition encapsulating OVA and GFP mRNA. They were perfused through the chip at 3 microgram per milliliter for 2 days. The results clearly showed GFP signal in myeloid subsets, specifically dendritic cells and monocytes, with no expression detected in B or T cells. This demonstrates precise myeloid tropism in this humanized model. As the authors concluded, myeloid cells were competent at capturing and expressing mRNA vectored by lipid nanoparticles, enabling the assessment of responses to mRNA vaccines. This validates the LO chip as a relevant human-like model for mRNA vaccine evaluation, and our LNPs as the right tool to power that research. The following data was generated in collaboration with Carmel Therapeutics and JC Discovery, for whom we developed a custom LNP formulation dedicated to T-cell transfection. The experimental setup is straightforward. GFP mRNA encapsulated in our LNP formulation was tested at 2 doses, 0.5 and 1.5 micrograms on human PBMC. PBMCs were pre-activated with anti-CD3 CD28 for 48 hours, and GFP expression was assessed by FACS. The results were very compelling. Looking at flow cytometry plots, we can see a clear and dose-dependent GFP signal across all cell populations tested: T cells, CD4, CD8, B cells, NK cells, and NKT cells. At 1.5 micrograms, we can reach up to 80% GFP-positive cells for CD4 and CD8 T cells. B cells and NK also show strong transfection, while NK are less permissive, which is consistent with what is generally observed in the field. Critically, viability remains above 85% across all doses for both T cells and total leukocytes, confirming that our formulation is not only efficient but also well tolerated. This positions our LNP platform as a relevant tool for CAR T-cell engineering and other T-cell-based therapeutic approaches. Finally, let's talk about one of our most advanced developments: surface functionalization of LNPs for targeted delivery. We typically use 2 grafting options. The first approach uses maleimide LNPs. In this example, the customer provides an antibody with accessible amine groups. A SETA crosslinker is used to introduce a protected thiol group on the antibody. After a deprotection step, the free thiol reacts selectively with the maleimide group displayed on the LNP surface, a reaction known as a SETA-maleimide conjunction. This is robust and well-established thiol-maleimide chemistry, particularly suited for antibodies and proteins with accessible cysteine residues. The second approach uses azid LNPs. Here, the antibody is modified with a DBCO group using a NHS-DBCO cross-linker. This DBCO group then reacts spontaneously and specifically with the azid group displayed on the LNP surface, proceeds without catalyst, and is fully biocompatible. Both strategies allow for efficient and stable conjugation of the targeting moiety to the PEG layer of the LNP, without compromising particle integrity or cargo encapsulation. And so, for applications requiring cell type specific delivery, we offer two complementary conjugation strategies to functionalize the LNP surface with targeting agents such as antibodies, nanobodies, or peptides. Maleimide chemistry and azid-click chemistry rely on grafting the targeting moiety onto functionalized PEG layer at the surface of the LNP. Maleimide approach is more appropriate for proteins or peptides that naturally or artificially contain free cysteine, as opposed to azid that requires DBCO modification of the molecule of interest. With these two tools, we can adapt to any situation. The choice between maleimide and azid chemistry typically depends on the nature and accessibility of the reactive groups on your targeting molecule. Our team is available to guide you toward the right strategy for your project. In summary, our LNP platform is fully adaptable to your system and your model. Whether you are working on mRNA therapy, genomic addition, vaccination, CAR T cell engineering, or any other nucleic acid-based application, we have the formulation expertise to support you. Our platform offers compatibility with any type of nucleic acid and small molecules, adjustable size and zeta potential, a library of more than 50 proprietary ionizable lipids with available licenses, fluorescent labeling for trafficking and imaging applications, and surface functionalization with targeting agents, whether peptides, antibodies, or other ligands. All of this is backed by rigorous quality controls and a team of experts ready to work with you from design to final characterization.
Thank you everyone for your attention. We will now go to the Q&A session. So we have a first question. Do we offer GMP grade LNPs? And if not, is that something that we could support for a clinical stage program?
No, not yet. This is not yet in our pipeline of in-house GMP production. However, this doesn't have to be a blocker. We can work with you to externalize or outsource the GMP manufacturing as soon as your program is ready, and we can of course externalize the production if it is needed.
Okay. Another question is for the DIY LNP kit, what formulation process do we recommend? And would it be possible to purchase only the ionizable lipid?
We recommend to use, we can support both either microfluidic or jet mixing approaches. And also there is a protocol that we have developed for end mixing procedure that we can send to the customer. And the best recommendation, depending on your application, is always to contact our technical service. And our formulation lab is always happy to get in touch and to make a call to discuss about your specific project. And now, considering the purchasing of lipids, I think you're...
Yeah, so for the ionizable lipid alone, I will have to check with the legal department because I think there is some IP involved. So I don't know if we can send only the ionizable lipid, but I will check and get back to you. We have another question for small molecule encapsulation. So what is the feasibility limits when it comes to encapsulating small molecules?
So this, it depends really on the case-by-case basis and on the hydrophilic and charge of the peptide and the molecule you want to encapsulate. We have, to give you a concrete example, if you got a highly hydrophilic and very charged, very positively charged molecule, you will reach the limit of the lipid matrix of the LNPs. So it will be difficult to encapsulate that, but this shouldn't be a concern because we have other solution for this. We can always use liposome to deliver the cargo, even if it is out of the limits of the LNP encapsulation. There is always a solution that we can find.
Okay, thank you. I can see that we have other questions. Everything will be answered by email because we don't want this session to be too long, but don't worry, everyone will get an answer. Thank you very much for your time, and please stay until the satisfaction survey. Thank you.
Thank you.